Can you see me?
Yeah.
Okay, great.
Good morning, and thank you, Debyani, for accepting our invitation. Happy New Year to you and your family. It's a great pleasure to see you, and looking forward to your presentation. Debyani Chakravarty is associate attending molecular geneticists at Memorial Sloan Kettering Cancer Center in the Department of Pathology and Laboratory Science, Medicine, and she's a key contributor and leader for the main OncoKB annotation, which is widely used at MSK Memorial Sloan Kettering Cancer Center. The patients. It's a privilege to hear the update from Debyani on OncoKB. Welcome, Debyani.
Hi, Semanti. It's so good to be introduced by my friend and colleague, Shimanti. So hi, everyone. I'll just get started. So first, I'll briefly describe the precision oncology landscape through the lens of OncoKB. And so it's not strange to any one of us on this call, but last year we documented on a granular level the progress of OncoKB. So back in 1998, the first precision oncology therapy was introduced. It was Herceptin for HER2-positive breast cancer. In that first decade and actually 15 years, the cost of sequencing was dropping precipitously, and we were getting massive landmark discoveries in the precision oncology field. Namely, BCR-ABL was connected as a predictive biomarker for response in chronic myelogenous leukemia to the targeted therapy imatinib in a retrospective study by Will Pao. EGFR exon 19 deletions, as well as L8 mutant L8 EGFR L858R was discovered in patients that responded to gefitinib and erlotinib. And then in 2010, 2011, BRAF V600E was discovered in patients' samples of melanoma, colorectal cancer, and lung cancer by the Stratton group in England. And then in the last decade, we've really seen this proliferation of biomarker discovery as well as consequent drug development that has really just had a spike in 2020 during the pandemic, where about 8 first-in-class FDA-approved precision oncology drugs were approved and a series of follow-on therapies. Today, 44% of all drugs in cancer treatment are precision oncology drugs. So almost half of all oncology drugs approved by the FDA are in fact precision oncology therapies that require biomarker testing in order for the patient to be treated by said drug. In addition, we have 9 tumor-agnostic precision oncology FDA drug approvals. This started in 2017 with the approval of checkpoint blockade therapy pembrolizumab for MSIH tumors. It was the first tumor-agnostic approval, and as recently as a couple of years ago, we had a groundbreaking approval of trastuzumab deruxtecan for HER2-positive cancers across all cancer types, as long as they present with 3+ immunohistochemistry. In the study that we executed last year, we also took a single patient cohort of about 47,000 samples from 42,000 patients across 66 solid patient tumor types. And we annotated it with OncoKB version 2017 and compared it against OncoKB version 2022. What we found was that there was an enormous, the same patient tumor samples, which as of 2017, only 7.7% carried a level 1 biomarker that was indicative of an FDA-approved precision oncology drug. That increased by 22.5% to 30%. This includes the 10% for TMB high, 9.2% for TMB high tumors that indicated pembrolizumab, but then that's still another 20% that were biomarker-based approvals. And we also saw an almost halving of the 44% of samples or 45% of samples that carried non-actionable mitogenic drivers. And so it essentially almost doubled the total actionable number of biomarkers. In more recent work, we recently published that we saw that from 1998 and more recently from 2012 to 2023, the likelihood of a patient getting matched with an FDA-approved precision oncology drug therapy increased for all patients. However, it didn't increase equally among all patients. So for patients of European ancestry from 2012 to 2023, we saw a 9-fold increase in clinical actionability for precision oncology versus only a 6-fold increase for patients of African ancestry. For patients of East Asian ancestry, the fold increase was 8.5, and for patients of South Asian ancestry, the increase was 6.8-fold. So that is the landscape in which precision oncology knowledge bases currently sit. And so just to kind of take a step back, where does the knowledge base sit in the precision oncology workflow? So precision medicine in cancer care has always been to use molecular profiles to identify clinically actionable driver alterations in patient tumor samples. And this starts with a patient tumor biopsy. It then progresses with mutation identification. So tumor biopsy and the accompanying normal sample is sequenced. The two samples, the normal and the tumor biopsy, are compared through bioinformatic analysis, and mutations, copy number alterations, and translocations are identified that are present in the tumor sample but absent in the normal sample. This is followed by mutation interpretation, which is case review and sign out by pathology, where knowledge bases such as OncoKB can assist in the interpretation and consequent clinical decision by the oncologist. Now, why is a knowledge base useful? This isn't new for many of us, but just as a level set, to reliably identify a clinically actionable variant, multiple cancer databases such as cBioportal, Cancer Hotspots, and so forth. The information resides in FDA drug labels, which specifies the exact biomarker in the drug label indicative of a specific precision oncology drug in a specific indication, or the NCCN conference proceedings, as well as the scientific literature. All of this information that resides in multiple unstructured databases have to be used in order to make the best clinical decision. And a knowledge base such as OncoKB integrates all of this information, and importantly, puts it in a programmatic fashion and allows this information to be programmatically accessed. Now, OncoKB is not the only precision oncology base out there. Of course, we know CIVIC by WashU, Cancer Genome Interpreter by Valdebron, and the very first cancer precision oncology knowledge base, My Cancer Genome, that was initiated in Vanderbilt University. But I'm going to go into OncoKB. I'm the lead scientist for this. So the history and a little bit about its current state. So in 2015, we started working on OncoKB in 2014 to be MSK's institutional knowledge base. In 2015, we defined the therapeutic levels of evidence, and in 2016 at ASCO, we released OncoKB through the website launch, as well as annotations being available through the public cBioportal interface. It was the secret sauce for OncoKB is the fact that it's powered by the clinical expertise of MSK physicians and physician scientists. We have a clinical genomic annotation committee comprised of 54 members of the clinical leadership at MSK. They respond to OncoKB emails that specifically to content requests that need to be included into OncoKB. In 2017, we published our first paper in JCPO, and OncoKB content was released institutionally via the MSK Impact Solid Tumor Reports. It is integrated as an annotation layer in cBioPortal and Empath and used to annotate, actually now it's 17,000 MSK patients sequencing reports annually. OncoKB is also currently used in MSK Impact liquid tumor reports. In 2020, we introduced licensing of OncoKB content to allow OncoKB to be self-sufficient. So licensed users are across the globe, both in academic and commercial, as well as hospital settings. And in 2021, October 7th, OncoKB was the first and still is the only somatic human cancer variant database to receive partial FDA recognition. And then recently we published our cancer discovery paper, and we're introducing germline content as well as integrating OncoKB into EPIC. So OncoKB has multiple layers of oversight and governance. We have an external advisory board comprised of leaders in precision oncology. It includes folks from MD Anderson, Princess Margaret Cancer Center in Toronto, Dana-Farber, as well as Johns Hopkins. Importantly, we have extensive engagement from the MSK clinical expertise. Again, as I mentioned, our clinical genomics annotation committee has disease management team leaders from prostate, breast, from the germline space, and so forth. And then we have a key OncoKB faculty, of which I am included, Nikki Schultz, the director of the Cancer Data Sciences Initiative, Mike Berger, the chief of clinical computational diagnostics, Mark Ladani, and David Solit. We have an incredible curation and software engineering team, and this really is what powers the engine that is OncoKB. Essentially, the curation is done by expert translational cancer biologists, and the software engineering is engineered by a team of software engineers. Importantly, OncoKB is currently a hierarchical database. Any clinical implication that links a targeted therapy to a cancer mutation has to be curated under a gene, mutation, and tumor type structure. We have a curation platform or a curation interface, which allows our curators to enter this information in a very structured way. And this allows for programmatic extraction of the data through our API. And as I mentioned before, it is the first and only somatic cancer variant database with FDA partial recognition, which covers the annotation of variants curated in OncoKB with an FDA level of evidence and credentials the robustness and transparency of OncoKB processes involved in variant evaluation and interpretation. This data is available through our website and ensures that test developers can use OncoKB data to support the clinical validity of tumor profiling tests in pre-market submissions. This is a screenshot from our website that displays the FDA-recognized content. We are not associated with any single companion diagnostic. We cannot be used to assess the analytical validity of a companion diagnostic. However, we can be used to assess the clinical significance of those biomarkers for any companion diagnostic, and this is available on our website. OncoKB, the levels of evidence were first published in 2015 May. The AMP-ASCO-CAP variant tiers of evidence was published in August. However, OncoKB levels of evidence map one-to-one to the AMP-ASCO-CAP levels of evidence. And of course, as part of our FDA recognition, we also map to the FDA levels of evidence. OncoKB is also the first and only somatic cancer knowledge base that is included in 2 of the NCCN solid tumor guidelines. This includes the NCCN for non-small cell lung cancer, where OncoKB is referred to for HER2 mutations, as well as, I believe, ERB2 mutations, as well as one other gene mutation, I can't remember. And it's also included for BRCA mutations in the uterine sarcoma NCCN. And lastly, OncoKB is one of 3 knowledge bases to have been identified as a valid and reliable source by the Medicaid Advisory Committee, Novitas, for CMS submitted local coverage determination. So how does one access OncoKB? So the first place is to go to our website, onkokb.org. Here we list all our 903 genes as well as 7,900 odd alterations. We have multiple overlapping levels of evidence. All of that information is available in a structured format. We also include 3 diagnostic levels for hematopoietic disease as well as prognostic levels. Additionally, OncoKB is integrated, as is VICC, as you see here, as an annotation layer in CBio portal. And if you hover over the OncoKB icon, you have access to the therapeutic implications as well as the biologic references that essentially establish the functional data behind the gene variant. And lastly, as VICC knows, OncoKB is programmatically accessible via its API, and one can annotate data files with an OncoKB annotator. However, you do need registration with the OncoKB website to use the annotator, and it has to be an academic effort. Otherwise, licenses are paid licenses through commercial entities for commercial entities are available. For clinical use in 2014, this is a sample signed out molecular pathology report that clinical oncologists were receiving after ordering MSK impact sequencing. Here you do not see the fact that there is an inactivating there's an inactivating TSC2 mutation, which at the time I'm just going to go back to my pointer, which at the time would have indicated the use of an mTOR inhibitor that was in clinical trial. Now, OncoKB, actually as of 2017, OncoKB is integrated as an annotation layer in MSK's sign-out platform that molecular pathologists can use the OncoKB information to sign out any MSK impact sequencing report. Automated OncoKB annotation is also included for it's actually gone up to about 18,000 MSK impact clinical sequencing reports per year, as shown here. Here we see that this is a bladder tumor sample that has high TMB, and therefore level of evidence by OncoKB is 1. And you'll also see a series of other levels of evidence for other mutations. The blue icon here designates that these mutations are drivers. The fire icon means that it is statistically recurrent. And then you have different summaries for, provided in the MSK impact clinical report. The treatment implications, the last sentence here is approved by our clinical genomics annotation committee. In terms of content development, in a study that was published in JAMA, I believe in 2017 or 2018 by Diana Mandelker et al. Here at MSK, we noted that of the 1,000 patients that had consented to their germline being sequenced, about 10% of those samples had a potentially clinically actionable finding in the germline setting. So it was clearly very important that the burden of all the 18,000 clinically OncoKB annotated MSK impact sequence reports have some germline variant annotation available for the molecular pathologist or clinical oncologist, and the burden doesn't fall onto the clinical genetics service. There's a subtle difference between germline versus somatic. So for example, here we're showing a breast cancer sample that has an inactivating BRCA mutation, which is considered level 3A for talazoparib and olaparib based on the current drug labels for these two drugs. So if in the somatic setting, it would be considered level 3A. However, FDA approved olaparib for germline BRCA mutated metastatic breast cancer, and this happened in 2018. So this would, if a BRCA inactivating mutation was found in breast cancer, it would, in the germline setting, this would be a level 1, not a level 3A. And so we've decided to introduce germline content. However, germline and somatic variants have different interpretations, implications, and treatments. In the somatic setting, oncogenicity is based on functional and clinical evidence. The majority are protein coding changes that arise primarily in the tumor. They're non-inheritable, and the implications are mostly binary. It's either oncogenic or non-oncogenic. In the germline setting, however, it's a little more complicated. The pathogenicity is defined by population data, family history of cancer, and et cetera, and not just the functional characterization of the mutation itself. Non-protein coding changes can be pathogenic, and it's present in every cell of the body. Therefore, there are treatment implications. There are family implications because these alterations are inheritable, and the spectrum of implications for genetic risk. So for the OncoKB germline project, we use the ACMG framework to classify pathogenicity, and we housed the pathogenic and likely pathogenic variants in OncoKB. We assigned germline alterations with recognized treatment associations based on an appropriate therapeutic level of evidence. And this was work that OncoKB published in collaboration with Sophia Stadler here at MSK in a JCO paper 4 years ago. And we provide structured and accessible data to store the MSK germline knowledge and make it a combined germline somatic reporting feasible. We house the genomic indicators in EPIC and allow OncoKB clients to accurately annotate NGS results in the context of either germline or somatic settings. This will be released at the end of this quarter or the beginning of next quarter. Essentially, you will be able to toggle between germline and somatic data. You can understand the genetic risk and genomic indicators associated with the gene, and you can view the annotated pathogenic and likely pathogenic alterations either at a C-dot or P-dot level. Variant interpretations curated by the Diagnostic Molecular Genetics Service here at MSK will be standardized by OncoKB and provided through our website. And the pathogenicity link out to ClinVar will also be provided. You can view the genomic indicators associated with a specific germline variant. So this is our timeline. It's probably going to be closer to the end of February, beginning of March, where we will release the germline content and the tier 1 genes. Tier 2, we anticipate in the summer, and then tier 3 by the end of this year. So where do we go from here? In addition to the germline content, as we currently report individual mutation-based annotations, so copy number gains and losses, indels, point mutations, translocations, and simple structural variants. In the future, what we're working towards creating is being able to include allelic configurations of structural variants like focal amplifications, copy neutral LOH, and so forth. We want to be able to annotate the whole genome and exome, but this will require some AI-based work. The most important, I think, development that we're really striving to figure out is the co-mutation annotation, being able to allow for different mutations to speak to each other, like resistance mutations and so forth. So this is where we are moving forward to. And I will end here. This this work takes a village. It's not just the OncoKB team. Then we're very dependent upon the OncoKB faculty, Nikki Schultz from CDSI, Mike Berger, the Chief of Clinical Computational Diagnostics, and Mark Ladani, Chief of Molecular Diagnostics, as well as Dave Sollett, who's provided instrumental advice through the lens of the Kravis Center for Molecular Oncology. We have a very small but a very mighty team. We have the scientific content management team that is essentially led by Sarah Soonholtz and Mariah Nissan, senior scientists on the team, as well as a fantastic team of software engineers that allows our API to be nimble, to include rules as well as exceptions to those rules, as well as our strategic initiative and knowledge system partners. The key is the codification of our Clinical Genomics Annotation Committee. These are clinical and scientific experts in the field of precision oncology within their specific disease management teams and our external advisory boards. So with that, I will end, and I'm happy to take any questions.
Thank you, Debyani. It was a great talk. I'll open floor for the question. Anybody has a question, please go ahead. We have enough time for discussion in case if anybody is interested.
Hey, Debyani, it's Wes.
Hey,
Great
Wes.
presentation.
Thank you.
I was just curious. So I know that you guys did all the mapping for the therapeutic response data back to the AMP-ASCO-CAP classifications. Do you guys have a timeline or plans for doing that mapping for the prognostic and diagnostic criteria as well?
Yes, I know this has been an ongoing discussion. We have so many projects for priority, particularly because of our commercialization efforts. We are currently undergoing sort of a revamping of our heme on which the diagnostic and prognostic mappings will depend. The thing is, we are transitioning to Epic, so that's kind of it's thrown a wrench into our plans. So we do anticipate right now where, it would most likely be end of first quarter in 2026.
Okay, thanks.
Thanks so much, Wes.
Hi, Debyani.
Hi, how are you? How are you?
Thanks for the talk. It's a pleasure meeting you. I'm familiarized with your work in the OncoKB.
Thank you.
I'm from Brazil. I work in Brazil in a clinical laboratory.
Wonderful.
With cancer genomics for a long time, and we do use a lot of OncoKB as a very trustful source of information. I want to ask you a question. Since we've been doing this cancer genomic variant classification for a while, we didn't have a consensus on variant classification until recently, right? We have 2 years ago a consensus of ClinGen, VICC, and CGC for variant classification. So we were in the past using most of OncoKB rational for the SOP for variant classification. But now we transition to more of this new form of classification.
Sure.
My question for you is how satisfied you are with the variant classification you guys have been using for a long time. If you see any room for updates, how do you see this new classification? I would like to get your opinion on that.
Sure. Just for the others on the call. So I believe Paulo, as he mentioned, in a couple of years ago, I think in 2021, 2022, Peter Horak, et al., Dimitri Sonkin, ClinGen, essentially it was a consensus built between ClinGen, VICC, and CGC. We published a point-based point-based system where oncogenicity has to meet certain criteria, and then on the basis of that, it's likely oncogenic, known oncogenic, and so forth. And OncoKB's version of variant classification is not point-based. It's essentially, it has to meet various criteria to be able to. And the question is, do we switch from the version that we have been using to the point-based system, on which I was an author on that point-based system. Right now, I can tell you that we are sticking to our current system, and the reason being, one, we've curated 7,900 variants, so to retrospectively go back and curate all of those variants and put them in a point-based system will be an enormous feat that we don't have the bandwidth to currently do. So that's kind of a practical reason. The second reason is a little more ambiguous in the sense that it's, while clinical actionability, it's important to define in discrete buckets, the oncogenicity is not as easy to be able to kind of bucket into a certain point level. Certain mutations are resistant, but not necessarily oncogenic. And so we don't want to include a framework that is fairly rigid in terms of the point-based system to categorize mutations when, in fact, if you categorize it as benign or non-oncogenic, then they don't get the necessary treatment implications. And so we try and stay away from that system and give ourselves a little bit of wiggle room when it comes to defining the oncogenicity or the treatment implications of a variant. Does that make sense?
Yeah. All right, thank you.
Yes, you're very welcome.
Hi, Debyani. This is Shobit Barua. I'm from West Virginia University. Thank you.
Oh, great. Hi.
Great talk. I was wondering about the germline part. So if I'm trying to understand, when you were dealing with the germline variants, or in the OncoKB, we are only including pathogenic and likely pathogenic, or you are also including VOUS? And if you're including VOUS or likely pathogenic, as time evolves, we know in germline that it can change the classification.
Sure.
How you're planning to deal with those kind of situations?
Yeah, that's a great question. So the germline content, we are literally getting data dumps from our diagnostics molecular genetics team. And so they curate every day the variants, they change the variant classification as needed, and we're creating a pipeline from their specific individual-based curation to OncoKB, which will be in a de-identified space. So if there are any changes, that will happen through a basically monthly update, and then we will show those changes on our website. And to the first part of your question, we're only sharing pathogenic and likely pathogenic variants. We're not sharing the benign or VUSs. If we do have VUSs, there will only be a link out to ClinGen. We're not trying to ClinVar, sorry. We're not trying to really go into the interpretation of that at all.
Thank you.
Sure. Yes, we do have date stamps. All of our data has a date stamp. So if you download the data, there will be a date stamp on the data. That is correct.
Any more questions? Debyani, I have more of a future direction question for you and also for the team. You said now we are going towards more whole exome, whole genome annotation, and how I'd just like to know your thoughts on how you're thinking about applying for using the AI into the interpretation, because this is a large, huge burden for the team. So what's your thoughts, and what is your plan for doing the AI integration into whole exome, whole genome?
Yeah, that's a really, it's a brilliant question. You're absolutely right. So David Soll at our Center for Molecular Oncology, he's been asking for whole genome annotation. And he literally asked us to manually annotate all 22,000 genes in the genome. And it became very quickly obvious that that's not possible. That's not possible from a manual perspective. So AI is definitely a resource that we are currently investigating. I think for whole genome, for whole exome annotation, there are two real stages. You've got 22,000 genes approximately in the genome. The first step is to figure out which of those genes are in fact cancer associated, and what are the criteria that you're using to define it as a cancer associated gene. And then the second level of curation is, okay, if it's cancer associated, is it a tumor suppressor or an oncogene or both or neither? Like, how are you defining what that cancer associated gene is functionally? And whether does the binary definition of oncogene and tumor suppressor still in fact apply when you're looking at this massive spectrum of genes? Initially, I had thought pretty naively that we could just identify those cancer-associated genes and manually curate them. That is no longer the case. So what we're trying to do is at first at least define what those buckets of cancer-associated genes are. Second, train AI, and we're going to compare a couple of samples, a couple of AI engines, like there's Llama, there's ChatGPT, and so forth. Train them on OncoKB data, and then use that as a predictive model. But it's definitely a very long-term vision, because even when you use AI to classify or to identify cancer-associated genes, there has to be sanity checks to say this is in fact what the AI is telling us to do. So we're certainly using AI to speed up our workflow, but it's still it's at least a 2-year long-term vision, not something we can report by the end of next quarter or anything like that.
Oh, thank you. This is great.
Thank you. Thank you.
Any other questions? Else we can just. Okay. Looks like great. This is a great talk. Thank you so much. And thank you, team, for everybody for joining us. See you again, Debyani. Take care. Thank you.
Bye, Shamanti. Bye, everyone.
Thanks
Bye,
all.
Paolo.